Guys, do you know who is this? Abraham Lincoln? Yes. Was Abraham Lincoln the 16th President of the United States affected with genetic disorder? Because several writers have speculated that he had Marfan syndrome.
I think that idea is based on his appearance and the reported Marfan syndrome in a distinct relative.
Was his physical appearance something to do with Marfan syndrome?
Photographs, written descriptions, and medical reports are available to provide ample information about Lincoln's appearance. He was 6'4" tall and thin, weighing 160 to 180 pounds. For almost his adult life, he has long arms and legs with large, narrow hands and feet.
Contemporary descriptions of his appearance indicate that he has loop-shouldered, loose-jointed, and walked with a shuffling gait. And in addition, he wore glasses to correct a vision problem.
His appearance and ocular problems are suggestive of Marfan syndrome. Marfan syndrome is a genetic disorder that affects the connective tissue of the body, resulting in a tall and thin individual, blood vessel defect, and shifted in the lens.
Others strongly disagree with speculations, arguing that his length, extremities, and proportion of his bodies are well within the normal limits of a tall, thin individuals. In addition, although he had vision problems, an examination reveals that he was farsighted, while those with classical Marfan syndrome are nearsighted.
And lastly, Lincoln shows no outward signs of problems with major blood vessels, such as the aorta.
Pea plants were selected by Mendel for two primary reasons. First, they can be crossed in all desired combination. Second, each cross is likely to produce large number of offspring. In particular, number of offspring is important in determining the mode of inheritance. Humans as a subject for genetic analysis neither satisfy these criteria.
Compared to the progeny that can be counted in a single cross with peas, humans produce very few offspring, and this usually represent only a portion of the genetic combinations that exist. If two heterozygous pea plants are crossed, 3/4 of the offspring will express the dominant allele B, while the recessive allele will be expressed in the remaining 1/4 of the progeny. Mendel was able to count hundreds and sometimes thousands of offspring from such a cross and to record progeny in all expected phenotypic classes, establishing a ratio of 3:1. As a parallel, consider two humans, each of whom is phenotypically normal. Suppose this couple has two children, one of whom is a son affected by a genetic disorder. The ratio of phenotypes in this case is 1:1. This makes it difficult to decide whether the trait is carried on an autosome or a sex chromosome, and whether it is controlled by a single gene or by two or more genes.
Good day everyone, my name is Ronalyn F. Tatuin, and today I'm going to discuss pedigrees and common patterns of inheritance in humans. So first, let's talk about pedigrees. An example earlier demonstrates that Mendel was able to count hundreds and sometimes thousands of pea plants offspring in a single cross. On the other hand, in humans, we cannot do such crosses. So in order to determine the mode of inheritance in humans, we map the trait or we use pedigrees.
So here is an example of pedigree. Pedigree uses standardized symbols. In a pedigree, circle represents females, and square represents males. The line between them represents mating, and this vertical line connects parents to offspring. Now, This shaded shape represents the trait, meaning to say that an individual exhibits or manifests the trait. The Roman numerals here represents the generations, and the Arabic numbers represents the number of individual in a generation. So here are other symbols used in pedigree.
So now, the question is, how would we know how a trait or traits passed on or inherited? We have several patterns, and I'm going to discuss 5 common patterns of human inheritance. So the first pattern that we'll be going to discuss is autosomal recessive. So we have 3 basic guidelines. First, the trait is located on non-sex chromosomes or autosomes. So if the trait is found between first and 22nd pair of chromosomes, the pattern of inheritance is autosomal. So how would we know by just looking at it? So if both sexes are equally affected, and male and female may transmit the trait. And second is, only homozygous recessive may exhibit the trait. So in result to that, trait may skip generations. Third is, consanguinity may be present. Consanguinity means mating between close relatives. So the second pattern that we'll be going to discuss is autosomal dominant. We have 2 guidelines. Just like in autosomal recessive, the trait in this pattern is located on non-sex chromosomes or autosomes. So meaning to say, both females and males are equally affected and may transmit the trait. Second is, only homozygous, dominant, and heterozygous individual may exhibit the trait. And in result to that, the trait does not skip generations. So next is X-linked recessive inheritance. So the trait in this pattern is located on X chromosome. So that's why it is called X-linked recessive inheritance. So both sexes can be affected, but take note of this. Males are more often affected than females. Why? Because it only takes 1 copy of recessive allele for them to manifest the trait. Unlike in females, it takes 2 copies of recessive allele. Another is, father to son transmission is never possible because since males have 2 kinds of chromosomes, X from mother and Y from father, the father only give Y chromosome to their son. And since we're talking about X-linked recessive trait or inheritance here, the father to son transmission is never possible. X-linked dominant inheritance. The trait in this type of inheritance is located on X chromosome. Both sexes can be affected, but females are more often affected than males. Affected males must come from affected mothers, because males get their X chromosome from their mothers. All the daughters, but none of the sons of an affected father are affected, because the Y chromosome of the father will pass on to his sons. For a female child to be affected, the father or the mother must be affected. And in this type of inheritance, the trait does not skip generations. And last is Y-linked inheritance. In this type of inheritance, the trait is located on Y chromosomes. And since females don't have Y chromosomes, only males are affected. Since only males are affected, this trait is passed on from father to son. And lastly, in this type of inheritance, the trait does not skip generations.
Hi, I'm Yvette, and I'm going to discuss about inborn error of metabolism and cytogenetics. First, let's talk about inborn error of metabolism.
About 1 out of 1,250 newborns is expected to be born with metabolic disorder, or also called inborn error of metabolism. It is inherited and is passed down unknowingly by parents who've not been screened before and may carry the defective gene copy. Let's go through some basics of genetic inheritance. All of us have two copies of a gene, one from each parent. Usually, if only one copy is defective, the gene can still function normally. However, if both copies are defective, the gene will not function properly, resulting in a disorder. In case of an inherited metabolic disorder, the person lacks a particular enzyme to perform normal metabolic function. A newborn with an inherited metabolic disorder lacks certain enzymes to break down milk properly, including breast milk from the mother. The affected baby becomes sick with various symptoms. For instance, poor feeding, vomiting, seizures, weak muscle tone, developmental delays, and lethargy. Sometimes, the symptoms may not appear at birth or may appear only when triggered by a certain situation. For example, period of fasting or illness. Some serious developmental issues and lifelong complications such as mental retardation, physical disability, or in some instances, death could occur if the affected baby was left untreated. The affected baby can live like any normal child if you detect and treat the metabolic disorders as early as possible. Treatment may include dietary management, supplement intake, and medication. The metabolic disorder can be screened via blood or urine. Urine collection is much easier and painless to baby. This simple and painless action can save your baby's life.
Now, let's proceed to cytogenetics.
Cytogenetics is an exciting, dynamic field of study which analyzes the number and structure of human and animal chromosomes. Changes that affect the number and structure of the chromosomes can cause problems with growth, development, and how the body functions. Chromosomal abnormalities can happen when egg and sperm cells are being made, during early fetal development, or after birth in any cell in the body. Changes to chromosome structure can disrupt genes, causing the proteins made from disrupted genes to be missing or faulty. Structural changes in chromosomes can lead to birth defects, syndromes, or even cancer. Alternatively, some chromosomal changes may have no effect on a person's health. Chromosome analysis or karyotyping is a test that evaluates the number and structure of a person's chromosomes in order to detect abnormalities. A regular human cell has 46 chromosomes, 44 autosomes which come in pairs, and 2 sex chromosomes which specify gender, XX for female and XY for male. A trained cytogeneticist can look for missing or extra pieces of chromosome. Chromosomes are arranged in pairs, from largest to smallest. This arrangement helps scientists quickly identify chromosomal alternations that may result in a genetic disorder. What happens when a person has something different, such as too many or too few chromosomes, missing pieces of chromosomes, or mix-up pieces of chromosomes? Sometimes, chromosomes are incorrectly distributed into the egg or sperm cells during meiosis. When this happens, one cell may get 2 copies of a chromosome, while another cell gets none. Incorrect distribution of chromosomes is called non-disjunction. Genetic disorders resulting from too many or too few chromosomes include Klinefelter syndrome, Turner syndrome, and trisomy 21.
I'm Jana Abonitalia and I'm going to discuss about the protein alterations due to mutation. What is mutation and its sources? The frame shift, missense, nonsense, and silence mutation, and the predisposition to allergy. What is atopy, asthma, allergic rhinitis, and atopic dermatitis? Protein alterations due to mutation. But first, what is mutation? A mutation is the permanent alteration of the nucleotide sequence of the genome. It is a change that occurs in our DNA sequence. So what causes the mutation? It's either the spontaneous mutation, which is naturally occurring, or the induced mutation that is caused by outside factors called mutagens. Mutations can alter the protein sequence. A frameshift mutation is a genetic mutation caused by insertions or deletions in the DNA sequence that shifts the way the sequence is read. Due to the triplet nature of the gene expressions by codons, the insertions or deletions change the reading frame or the grouping of codons, resulting in a completely different translation from original. The resulting protein is usually non-functional. For example, the group of codons ATG, GAA, GCA, and CTG, the protein that's encoded, it would be methionine, glutamic acid, alanine, and glycine. But then, in the second codon, the first one is deleted. The resulting DNA would be ATG, AAC, CAC, and GT. So the protein that would be encoded would be methionine, lysine, histidine, and question mark. Next one would be missense mutation, a point mutation in which a single nucleotide results in a codon that codes different amino acid. A type of non-synonymous substitution means it alters the amino acid sequence, contrasted with the synonymous which do not alter amino acid sequence. For example, the group of codons ATG, GAA, GCA, CGT, the protein that is encoded is methionine, glutamic acid, alanine, and glycine. Notice the second group of codons. The letter A in the third one is replaced or substituted by letter C. So the DNA sequence would be ATG, GAC, GCA, and CGT. The protein that is encoded would be different: the methionine, the aspartic acid, alanine, and glycine. So the glutamic acid is replaced with aspartic acid. The next one is the silent mutation. It is a substitution mutation that can occur in non-coding regions, or they may occur in exons. When they occur with exons, they either result as a synonymous substitution, which they cannot alter the amino acid sequence, or result in extension of an alternative amino acid with similar properties to original amino acid. For example, the group of codons ATG, GAA, GCA, CGT, the protein that is encoded is methionine, glutamic acid, alanine, and glycine. So the proteins that the protein would be the same in the next one, but the DNA sequence is different. The ATG, GAA, GCA, and CGT, still methionine, glutamic acid, alanine, and glycine would be its protein sequence. But in the next group of codons, the second group of codons, the third letter is replaced by letter G. So it's called silent mutation. Next one is nonsense mutation. It is a point mutation in a sequence of DNA that results to a stop codon. It differs with missense mutation, which is a mutation where single nucleotide is caused by substitution of a different amino acid. For example, the ATG, GAA, GCA, CGT, and the protein is methionine, glutamic acid, alanine, and glycine. Then the next DNA is ATG, TAA, GCA, CGT, where in the next or the second group of codons, the first letter is replaced, or the first letter G is replaced by letter T. So the protein sequence that is encoded is methionine and then a stop codon. Atopy is a predisposition toward developing certain allergic hypersensitivity reaction. It is a genetic tendency to develop allergic disease such as atopic dermatitis or eczema, allergic rhinitis or hay fever, and asthma. The signs and symptoms: the patients with atopy also have a tendency to have food allergies, allergic conjunctivitis. Atopic syndrome can be fatal for those who experience serious allergic reactions such as anaphylaxis brought on by the reactions in food and environment. So, the asthma. It is a common long-term inflammatory disease of the airways of the lungs. The symptoms include wheezing, coughing, chest tightness, and shortness of the breath. Asthma is thought to be caused by a combination of genetic and environmental factors. Environmental factors include the exposure to air pollution and allergens. The causes is still unknown, but the genetic factors is the certain genes identified or the family history. The next one is the allergic rhinitis or the hay fever. It is an allergic disorder characterized by an exaggerated immune response to pollen grains and other substances. It could be perennial, where it occurs all year around, or seasonal, that occurs during the time of the year. The hay fever is often inherited trait. Next one is the atopic dermatitis or the eczema. It is a term for a set of chronic skin conditions caused by inflammations. Atopic dermatitis is the most common type of eczema. Itching and rash can be triggered by many things: allergens like dust mites, harsh soaps, fumes, and chemical, weather changes, and stress.
Sir, and I'm going to tackle or discuss about the behavior genetics. What is behavior genetics? What are those disorders that we might get from our families and their environmental factors? And I'm also going to tackle about the screening test, especially for the newborns. Screening test that can help your baby from the disease called the fetal phenylketonuria and the symptoms that your baby might gonna suffer after delivery. So let's start. Behavior genetics, also referred as a field of scientific research that uses genetic methods to investigate the nature and origins of individuals' differences in behavior. So, behavior genetics is also called or also known as the scientific discipline by Francis Galton. Another aspect of genetics is the study of the influence of heredity in behavior. Many aspects of animals' behavior are genetically determined and can therefore be treated as similar to other biological properties. This is the subject material of behavior genetics, whose goal is to determine which genes control various aspects of behavior in animals. Human behavior is difficult to analyze because of the powerful effects of environment factors such as culture. Few cases of genetic determination of complex human behavior are known. Genomics studies provide a useful way to explore the genetic factors involved in complex human traits such as behavior. Single gene diseases caused by mutant alleles of a single gene. It has been discovered 2 wild characteristic single gene diseases include phenylketonuria, such as heart disease, schizophrenia, and depressions, are thought to have more complex hereditary components that involve a number of different genes. These diseases are the focus of a great deal of research that is being carried out today. Another broad area of activity is clinical genetics, which centers on advising parents of the likelihood of their children being affected by genetic diseases caused by the mutant genes and abnormal chromosome structures and number. Such genetic counseling is based on examining individual and family medical records and on diagnostic procedures that can detect and express abnormal forms of genes. Counseling is carried out by the physicians with a particular interest in the area or by specially trained non-physicists. What is behavior genetics? It is also called a psychogenetics, the study of the influence of an organism's genetic compositions on its behavior and interaction of hereditary and environment insofar as they affect behavior. The question of the determinants of behavior abilities and disabilities has commonly been referred to as the nature-nurture controversy that was being exiled by Francis Galton. The relationship between behavior and genetics or hereditary dates to the work of the English scientist Sir Francis Galton in 1922 and 1911, who coined the phrase nature and nurture. Galton studied the families of outstanding men of his day and concluded, like his cousin Charles Darwin, that mental powers run in families. Galton became the first to use twins in genetic research and pioneered many of the statistical methods of analysis that are in use today. In 1918, British statistician and genetics Ronald Aylmer Fisher published a paper that showed that how Gregor Mendel of inheritance, the law of inheritance, applied to complex traits influenced by multiple genes and environmental factors. Behavioral genetic research today focuses in identifying specific genes that affect behavioral dimensions, such as personality and intelligence, and disorders, such as autism, hyperactivity, depressions, and schizophrenia. So we have here the schizophrenia. Schizophrenia is a chronic and severe mental disorder that affects how a person thinks, feels, and behaves. This disorder typically appears during the adolescence or the early adulthood. The signs and symptoms of this disorder include false perceptions called the hallucinations. The person that can see things that cannot be seen by the others. Imaginary voices or delusions that are most common hallucinations in schizophrenia, but affected individuals can also experience imaginary visions, smells, or feelings of being touched, but they are not being touched. Strongly held false beliefs or delusions are also characteristic of schizophrenia. People with schizophrenia often have this decreased ability to function at school, work, and social settings. Disordered thinking and concentration, inappropriate emotional responses, erratic speech and behavior, and difficulty with personal hygiene and everyday tasks are also features of this disorder. The environmental factors involve such exposure to virus, mutation before birth, problems during birth, and the psychosocial factors. So let's proceed to the eating disorder. It describes illnesses that are characterized by irregular eating habit and severe distress or concern about body weight or shape. So this disorder, it also runs in our families. But the study finally pinpoints two genes that significantly increase a person's risk of developing anorexia and bulimia nervosa. Eating disturbances may include inadequate or excessive food intake, which can ultimately damage an individual's well-being. The popular or the most common factors is the anorexia nervosa. It is the male or female or an individual as a whole. It is the fear of gaining weight. Mainly people who has this disorder will fiercely limit the quantity of food they intake or consume. Even they are literally thin, they always insist to themselves that they are fat. So the next factor is the bulimia nervosa. Bulimia nervosa is characterized by repeated binge or indulging food, followed by behaviors that compensate for the overeating, such as force in vomiting, excessive exercise, or extreme use of laxatives and diuretics, the medicines that can make you lose weight. So the next one is the binge eating disorder. Binge is also called or also known as indulging. So frequently lose control over his or her eating. This is like bulimia. But it is not followed by the compensating behaviors, that taking medicines that can make you lose weight. This suffers from bad laziness. Cause of eating disorders. Biological factors include irregular hormone functions, genetics, nutritional deficiencies, and in psychological factors, the negative body image and the poor of self-esteem. The environmental factors, which are the dysfunctional family dynamic, professions and careers promote being thin. Aesthetically oriented sports, example, diving, ballet, the gymnastics, family and childhood traumas, cultural or peer pressure among friends, co-workers. Stress also is the environmental factors for eating disorders. What is drug addiction? Addiction is defined as a chronic relapsing brain disease that is characterized by compulsive drug seeking and use, despite harmful consequences. It is considered a brain disease, harmful because drugs can change the brain. They change its structure and how it works. These brain changes can be long-lasting and can lead to the harmful behaviors seen in people who abuse drugs. Addiction also is a lot like other diseases, such as heart disease. Both disrupt the normal, healthy functioning of the underlying organ, have serious harmful consequences, and are preventable and treatable, but if left untreated, can last a lifetime. So why do people take drugs? Generally, to feel good. Most abuse drugs procedure intends feelings of pleasure. This initial sensation of euphoria is followed by other effects which differ with the type of drug used. For example, with stimulants such as cocaine, the high is followed by feelings of power, self-confidence, and increase of energy. In contrast, the euphoria caused by opiates such as heroin is followed by feelings of relaxation and satisfaction. It also to be feel better. Some people who suffer from social anxiety, stress-related disorders, and depressions begin abusing drugs in an attempt to lessen feelings of distress. Stress can play a major role in beginning drug use, and we all know that. Continuing drug abuse or relapse in patients' recovery from addiction. And curiosity and because others are doing it. That is also factors. In this respect, adolescents are particularly vulnerable because of the strong influence of peer pressure. Teens are more likely than adults to engage in risky or daring behaviors to impress their friends, express their independence from parental social rules. So, Why do some people become addicted to drugs while others do not? As with any other diseases, vulnerability to addiction differs from person to person, and no single factor determines whether a person will become addicted to drugs. In general, the more risk factors a person has, the greater the chance taking drugs will lead to abuse and addiction. Protective factors, on the other hand, reduce a person's risk of developing addiction. Risk and protective factors may be either environmental, such as conditions at home, at school, and also in our neighborhood, or the biological. For instance, a person's genes. If you start out with low genetic predisposition or addiction, you can still end up with an addiction. But as I have tackled earlier, there's environmental factors that increase the risk of addiction. But now I am going to elaborate it more. Home and family. The influence of the home and environment, especially drug childhood, is a very important factor. Parents or older family members who abuse alcohol or drugs, who engage in criminal behavior, can increase children's risk of developing their own drug problems. Peer and school. Friends and acquaintances can have an increasingly strong influence during adolescence. Drug-using peers can sway even those without risk factors to try drugs for the first time. Academic failure or poor social skills can put the child at further risk for using or becoming addicted to drugs. So let us now proceed to the mood disorder. A psychological disorder characterized by the elevation or lowering of person's mood, such as depressions and bipolar disorder. Depression is often a family affair. Scientists, for example, have identified a gene that may be linked to bipolar disorder. In addition, they have found a common genetic mutation associated with a person's developing clinical depression when faced with traumatic events in their lives. Genetics. Although genetic factors may make a person more susceptible to mood disorders, a trigger such as serious medical conditions or psychological stress is often what causes a mood disorder to emerge at a specific time. So, does intelligence determined by genetics? Let's find out. But first, we have to define intelligence in humans first. Intelligence in humans is the intellectual prowess of a human, which is marked by high cognition, motivation, and self-awareness. Like most aspects of human behavior and cognition, intelligence is a complex trait that is influenced by both the genetic and environmental factors. Intelligence is challenging to study, in part because it can be defined and measured in different ways. Most definitions of intelligence include the ability to learn, to reason out, plan out, solve problems, think abstractly, and understand complex ideas. Many studies rely on a measure of intelligence called the intelligence quotient, which is the IQ. Researchers have conducted many studies to look for genes that influence intelligence. Many of these studies have focused on similarities and differences in IQ within families, particularly looking at adopted children and twins. These studies suggest that genetic factors underlie about 50% of the difference in intelligence among individuals, which makes only a small contribution to a person's intelligence. It is also strongly influenced by the environment. Factors related to a child's home, environment, and parenting, education and ability of learning resources, and nutrition, among others, all contribute to intelligence. A person's environment and genes influence each other, and it can be challenging to tease apart the effects of the environment from those of genetics. That's all for the behavioral genetics, and now let's proceed to the screening test for the newborn. Let us now proceed to the newborn screening test. So, what comes to our mind if we hear screening test? So let's define it. Are laboratory tests that help identify people with increased risk of condition or disease before they may be at risk so that preventive measures can be taken. Let's focus to the newborn screening test. Newborn screening test looks for developmental, genetic, and metabolic disorders in the illnesses are very rare but can be treated and caught early. Let's look for phenylketonuria. So what is phenylketonuria? It is a rare condition in which a baby is born without the ability to properly break down an amino acid or the phenylalanine. So, what causes this disorder? The causes of disorder is inherited, which means it passed down to the families. Both parents must have passed on a non-working copy of the gene in order for a baby to have the condition. Babies with PKU or phenylketonuria are missing an enzyme called phenylalanine. The hydroxylase, which is needed to break down the essential amino acid, which is the phenylalanine. Phenylalanine is found in the foods that contain protein. Without the enzyme, the levels of phenylalanine build up in the body, and this build-up can harm the central nervous system and can cause brain damage. So, what are the symptoms of this? Phenylalanine plays a role in the body's production of melanin, the pigment responsible for skin and hair color. Therefore, infants with the condition often have lighter skin, hair, and eyes than brothers or sisters without the disease. Other symptoms may include the delayed in mental, social skills. Second, the head size much smaller than the normal. The hyperactivity, seizures, skin rashes, and tremors, and even jerking movements of all arms and legs. If PKU or phenylketonuria is untreated, or if foods containing phenylalanine are eaten, the breath, skin, earwax, and urine may have a mousy or musty odor. This odor is due to the build-up of phenylalanine substance in the body. So how can you prevent this? An enzyme assay or genetic testing can determine if parents had carried all the way with PKU or the phenylketonuria. Embryonic villus sampling or amniocentesis can be done during the pregnancy to screen the unborn baby for PKU. It is very important that the woman with PKU closely follow a strict low phenylalanine diet, both before becoming pregnant and throughout the pregnancy. Build-up of phenylalanine will damage the developing of baby, even if the child has not inherited the defective gene. So for the conclusion for my report, don't neglect screening test, because there's a possibility that your baby will suffer PKU, and you better have a test or your baby will suffer. So that's all. Thank you for listening and have a nice day.